This is Mrs. Palmer Quay with an overview of the concepts in Chapter 16 on DNA technology. This is not a chapter that you have to read for my course, but since DNA technology is very important for our lives today, it has influences the treatments that are happening in medicine and in our food production, it's important for you to have some understanding of what's happening in this process. DNA technology is a various number of procedures that are used to recombine DNA from two different species. And so this recombining DNA also means that it can be called recombinant DNA technology. Some of the ways that we have mixed DNA together is to mix human DNA with bacterial DNA. This is what's involved in producing insulin for people who are diabetic. We've mixed human DNA with the mouse to provide a lot of mice that are important in research and trying to understand what genes do and how it contributes to human health. Viral DNA, or DNA from viruses, mixed with bacteria is how we produce many of the vaccines that we use to, again, keep us healthy. Jellyfish have contributed a particular gene to a number of laboratory animals. The jellyfish gene allows these animals to show up glowing green under UV light, and it is used mainly as a marker for another gene that's being tested. And then there's several different combinations that are used in the plant world. One is a bacterial gene for to cause disease to a particular insect pest, and so plants that come with this DNA in it are already resistant to that pest. DNA technology also is known as biotechnology or genetic engineering, and it started in the 70s and has really become a very important part of our world. It has influence all over the place. The basic process is that the DNA that we're worried about or chosen for whatever reason, the selected DNA, is inserted into another DNA molecule, and it's often into one of these circular bits of DNA called plasma. So in this little bit of DNA, this is the gene that for whatever reason the scientists want to have it expressed, and so it's inserted into another piece of DNA, and then that piece of DNA enters a host organism, in this case a bacteria, where the gene would reproduce itself, and then as it is expressed, that host organism has to be one that favors that gene, and then as the gene is expressed, it produces whatever protein it codes for, and then that protein is available for use. Why do we do DNA technology or recombinant DNA technology? Well, certainly an important reason is because it gives us substances that can be used to treat or cure human disease. I've already mentioned insulin. The various clotting factors that are for people who are hemophiliacs and do not produce enough of them on their own are now produced through genetically modified bacteria. Our vaccines are produced through this technique. We also, as I said, learn a lot more about diseases, about what happens if you do not have a gene working correctly by using animals that have a bit of human DNA. And so if any animal that has, or any creature, organism that has a bit of some other organism's DNA in it is known as a transgenic organism. So we have transgenic mice that are used extensively in research. And then in the agricultural setting, the choices have been made to try to produce foods that have a high level of a particular nutrient. That example of that would be something called golden rice, which contains the precursor beta-carotene, which makes vitamin A when someone eats it. It's a brand new product that's just starting to be introduced in parts of the world where children suffer blindness because not enough vitamin A is taken in. There are a number of commercially grown products that have pest or pesticide resistance built into the plant. Bt corn is an example. Bt is a bacteria that harms caterpillars, and so here is corn that will kill the caterpillars if the caterpillars eat them. Roundup Ready corn is corn or soybeans or beets that can be sprayed with Roundup, which is an herbicide, something that will kill plants normally. Well, it won't. You can spray it on the fields and it will kill the weeds, but not your crops if you use Roundup Ready crops. GM organisms also, or GM stands for genetically modified. GM organisms food organisms have been produced that have a longer shelf life. There's a tomato that has a shelf life of 45 days instead of 15. And then certain genetically modified products are involved in getting a higher production overall, either of a plant's yields or the bovine somatotrophin that is used to increase milk production in dairy cows is a genetically modified chemical. It's produced by genetically modified bacteria. So let's go over a few key terms. The first one I want to talk about is a plasmid. Plasmid is a small circular piece of DNA that's found in bacteria, and a few fungus also have plasmids. So here's on this diagram over here, here's our plasmid. You see it's separate from the normal bacteria chromosome, the DNA that is necessary to provide all the information this bacteria needs to grow and survive and stay healthy. So the plasmid has been picked up from the environment, but it now has been incorporated into this bacteria. And often on plasmids is where you're finding the antibiotic resistance that is a concern in medicine. Many of the bacteria that used to be treated with a particular drug, you no longer are able to get a kill from that drug. And the resistance, the genes that have that resistance are found on these small bits of DNA known as plasmids, which can be transferred from bacteria to bacteria very easily, and that's why antibiotic resistance can go through a bacterial population. The next key term is a restriction enzyme, and DNA technology would not happen if we didn't know about restriction enzymes. A restriction enzyme cuts the DNA in a very specific place, and there are different restriction enzymes that cut at different places. So this particular restriction enzyme makes a cut when in a place where this code is found. And so you see it now has split this double-stranded DNA into two sections, and that these two ends here are complementary because that's where it's been cut, and we call those ends the sticky ends. So what's important about restriction enzymes is that any piece of DNA that's cut with a particular restriction enzyme will stick to any other piece of DNA cut with that same restriction enzyme, even if they're from entirely different species or sources. So we've got one DNA here from one organism and another bit of DNA over here from another organism, but because they have been sliced open with the same restriction enzyme, these two ends are complementary or sticky, and through the right process of another enzyme being involved, can actually join these two pieces of DNA together. And so this is how we insert bits of DNA, a particular gene that is trying to be manufactured, or the protein that it makes is trying to be manufactured in bulk. This is how we put it into another organism in order to use this DNA technology. It's because the restriction enzyme will cut all DNA at a particular place, and then those divergent pieces of DNA can be bound together. Vector is the word we use to describe whatever thing we use that will put that piece of recombinant DNA into a host. Typically we're using viruses or plasmids as vectors. Plasmids generally can be taken into bacterial cells very easily if they are just in the outside environment. Bacteria seem to like to suck them up. Viruses, by the way that they work, actually inject their DNA into the host cells, and then that DNA co-ops takes over the host DNA and makes many, many little viruses until that cell bursts. So if you can get a bit of recombinant DNA into a virus and turn that virus loose, it'll just go about injecting it. The host organism is the organism that receives this recombinant DNA, and this little R in front of it is a way to abbreviate it. And that host organism then is the one that expresses the gene. And remember, when you express a gene, it just means that you make the protein that the gene is coded for. Along with using plasmids or viruses, there are other ways of inserting DNA into host organisms. We have microinjection with a tiny, tiny micropipette, which will put DNA directly into the cell. This is the technique we actually use with cloning when you're putting an entire nucleus into a cell. Electroporation uses pulses of high voltage electricity, which then triggers small openings in the cell membrane and allows the recombinant DNA to sort of slide in if it's in the outside environment. And there's also something known as a gene gun, which shoots high-velocity particles of gold or tungsten or other heavy metals that have been coated with DNA. And so they basically are shooting those into the cell, hoping that that little bit of DNA will be picked up and incorporated into generally it's bacterial, the bacterial cell itself. all methods of introducing this of recombinant DNA, any of these have actually a low efficiency. So after you've treated a selection of host cells, you have to have techniques that allow you to separate which ones actually have picked up the desirable gene and which ones haven't. So to just kind of summarize this in a picture form, we've got our DNA that has a gene in it that we want to get a little, we want to take it out and use it more specifically, and we've got a plasmid over here that's going to be the vector for carrying this gene. So through a number of techniques that you don't need to know about, we're able to isolate that gene and use the restriction enzyme to cut out the piece with these little sticky ends on either part. And the same thing, the plasmid itself is cut apart so that the sticky ends allow that piece of the gene to be, or that particular gene, that piece of DNA to be inserted into the plasmid. And then one of these techniques are used to have the plasmid taken up by the bacteria. And as it says in here, you know, some of the bacteria may take up the plasmid and some won't. You can see here's one that just did not do it. Because in the case of the plasmid, it's usually just inserted into the outside environment, the fluid environment the bacteria are living in, and then time is just allowed for the bacteria to pick it up or not. And then there is, again, some way to make sure that to separate out the bacterial cells that actually carry that gene. So in this particular example, it's a bacteria that when the cut was made here, you're actually cutting, when you're cutting this one apart, you are deactivating a particular gene, and so that allows you to then separate out which cells have had that gene deactivated in the bacteria and which one have not. If the bacteria gene has been deactivated, then you know that this other gene has been inserted. So some examples of things that we are now using DNA technology to produce. I mentioned human insulin, which is a hormone that allows your body to take glucose, sugar, out of your blood. If your pancreas breaks down, stops producing insulin, then you have diabetes. Previously, we got the insulin that was used to treat diabetes from extracting it from ground-up pancreas from farm animals, specifically cows and pigs. But because of that, people could have allergic reactions to the insulin because it wasn't human insulin, it was cow insulin or pig insulin. So we now have created a strain of E. coli bacteria that makes the parts of human insulin. There are two different proteins that are involved in making human insulin, so they are actually put together in the laboratory afterwards to make the functional hormone. But the bacteria are making the parts, and we can have lots and lots of bacteria in a little bit of space, and they can pump out lots and lots of insulin. This is very, very similar to the process that's used to produce vaccines, which I have a picture of on the next slide. In the vaccine, what you're doing is creating antigens, or you're triggering antibody production by creating antigens that identify something as a virus without that antigen actually being on the virus. So the DNA technology takes that little bit of identifying marker, sticks it in a bacteria that produces lots and lots of it, and then we can insert this marker into people's body. Our bodies will respond to that antigen and make antibodies, but there's no live virus there. There's not the rest of the virus machinery to give you smallpox or to give you polio or to give you measles or mumps, these many things that we get immunized against. And so in this case, again, the DNA technology makes a little piece of the virus that really counts so far as triggering the antibody production in our own bodies. Gene therapy is really still very much in its infancy. There are many different trials underway to use gene therapy. A few techniques have had success and are in a little more common use, but basically we're still beginning in this process. But here you see how the idea is that you are making genetically altered cells, so the cells are removed from a person. A virus is altered so it can't reproduce. This is your vector. The gene is inserted into the virus, and the virus is reintroduced into the cells so that inside the person's body we've created a new kind of cell that wasn't there before. They were missing a particular gene. This is where you use gene therapy to replace a gene that was missing. And so you're introducing cells that have that gene and will make whatever that gene needs to make. This typically is done for someone who is already born. It's done for the cells in the body, so it's a somatic cell replacement. As I said, we're just really beginning this process. There are some success stories that you can find, but we still have a lot to do in making this a very common way. It's nowhere near, the way that you would normally treat disease. We're just still really beginning. And then finally, there's a lot of recombinant DNA technology being used in agriculture. This is one area where I'm not quite so sure that I think all of it is a good idea. It's used to create produce that takes a longer time to ripen, so it can be shipped longer distances. As I said, it's used to provide insect or disease resistance in plants, to increase the nutrient content, as I mentioned earlier. And then there's some thought about making edible vaccines instead of injectable vaccines, but edible vaccines in plants for use in developing countries. There are concerns over genetically modified foods. These are being expressed and determining policy more in places that are not the United States. The United States, there's a very close relationship between the regulatory agencies and the money-making agribusiness corporations. And so simple things that you would think would be very logical, such as identifying what country produces our food. This is known as country of origin labeling. It took years to get that passed because there was so much resistance among the industry. The various food corporations did not want to tell where they were sourcing their food from to the American public. And so it's very hard really for Americans to get much information about our food. In the other parts of the world, people are able to get policies enacted that allow them to make wise, knowledgeable decisions about food because things are labeled or things are more carefully controlled than in the United States. So in other parts of the world, these are the big concerns that there has been action taken. Because you're putting DNA from one organism into another, there is a concern about allergic reactions potentially developing. To my knowledge, there's not been anything that has happened yet, but you could see if somebody is allergic to peanuts and never expects to see a peanut gene in their tomatoes, but a peanut gene has been used for some particular reason because it makes this tomato more profitable, then they might have an allergic reaction to the tomatoes because of the presence of the peanut gene. So that's the concern, that the foreign genetic material will make something cause an allergic reaction where it never was expected. There's also the concern about gene transfer to the bacteria that are normally residing inside our digestive tracts, because as I said, bacteria will pick up plasmids very easily, and bacteria in bacteria colonies spread antibiotic resistance through the community very easily. And so the fact that antibiotic resistance has been used as a marker in many of these initial trials of GM foods. So they'd use a fact that a bacteria was resistant to an antibiotic as a way of marking which one has picked up that gene because of what plasmid they put the gene on. That means that the gene that is put then into the food also contains that antibiotic resistance. And if that then gets into the person's digestive system and is picked up by the bacteria that normally reside in their digestive system, then it becomes possibility they could suffer from a disease that we could not treat them for. And then another really big concern is the fact that you're talking about plants that reproduce by pollen, and many of them reproduce by wind pollination, where vast amount of pollen is released. The corn, for example, is wind pollinated, and the pollen travels for miles. And so there may be outcrossing occurring with non-genetically modified varieties that are in the same area within several miles of the fields of whatever crop. And that will either cause the native species to be completely overrun because they are not able to combat the altered genetic makeup of the GM varieties, or just cause an overall decrease in genetic variability, which then makes us more susceptible to losing our food supply when some new disease comes along. So these are concerns that people have about genetically modified food. I personally would like to see labeling. As I said, there's no labeling right now legislated in the United States because there really has been a push by the food industry to keep everything secretive and quiet, and I'm not real thrilled about that. I would like to be able to know what I'm eating. I don't know if genetic modification will actually get as far as this cartoon suggests. But it certainly is something that is important for how we are treating our diseases and health and medicine today. It has become a true reality in our food supply, human food supply, and especially the livestock food supply, because corn and soybeans are the two big genetically modified crops, and they are the base for livestock feed. And so it's going to be something that's part of your life, and hopefully this video will help you understand things as you hear about concerns in the news or new developments in medicine.